Good morning, Christos.
Good morning. How are you?
Good. How are you?
All good. All good here.
Good.
Julia, are you ready?
Of course, I was born ready.
Exactly.
I love the optimism.
Right, we have 21 participants already, so we are live, so welcome.
Yeah, I can't see all of that, so you can let me know when we're ready to go, Lauren, because
Sure,
in presenter view I can't see all of that detail.
Okay, I'll let you know, and we can just give everybody a minute just to join, and then I'll give you a shout then. Thanks. We're just going to give a few more minutes for more attendees to join this webinar. Right, Megan, I think we can start now. I think we waited 2 minutes. Okay, and we can begin.
Thanks, Lauren. Good morning, everyone. Thank you very much for joining us this morning. My name is Megan Kotsia, and I'm the product manager for IDT at Whitehead Scientific. Today's presentation is jam-packed with information, so we will have a few minutes at the end for a Q&A session. So we ask that you put your questions in the comment box, and we will try and get to as many of these as possible after the session. Or alternatively, we'll be in touch with you to address your questions if we cannot cover it today. Please keep in mind that we still have another 2 topics in our seminar series, which is coming up. That's xGen NGS hybridization capture, your flexible solution for targeted sequencing, which will be hosted on the 20th of September, and xGen NGS amplicon sequencing, which will be hosted on the 27th of September. But back to today's session. Today, we are going to be talking about xGen NGS library presentation, library preparation, and xGen Normalase to accelerate your next breakthrough, which is going to be presented by Julia Duda from IDT. So IDT, or Integrated DNA Technologies, is a supplier of custom nucleic acids, serving the areas of academic research, biotechnology, clinical diagnostics, and pharmaceutical development. In 2021, IDT acquired Swift Biosciences, and they've gone on to expand their xGen NGS portfolio substantially. Our presenter today is, I just need to get my screen to change here. Here we go. Our today is a member of the field application team in Europe, and she provides support to research using IDT's range of xGen NGS solutions in the field. She completed her bachelor and master's degree in biotechnology at the University of Poland and received her PhD in molecular biology from the University of Freiburg in Germany. So we just want to say welcome to Julia today. Thank you very much. We also have Christos from IDT. So we have a range of experts online for anyone that has any questions today. So thanks very much, Julia. I'm going to hand over to you now.
Thank you very much for your kind introduction. I just need to share my screen, and for this, if you could.
There we go. Should be able to share your screen now.
Can you confirm you can see my screen?
Can see it.
Perfect. So without further ado, I will begin. Before I start, I just wanted to say a few words about our company. So you have already mentioned a few words about IDT. Just very brief history of IDT. We were founded by Dr. Joseph Walder from the University of Iowa in 1987. And IDT is the leading brand in DNA and RNA synthesis, but over the last 30 years, we have also expanded significantly our portfolio to cover more and more aspects of molecular biology research. Today, we are more than 1,500 associates across 11 locations. We have more than 1,300 active customers, more than 4,000 orders per day. Our headquarters and most of the production facilities are based in USA, but also in Belgium and Singapore, and we have global sales presence. Our expertise in DNA synthesis allowed us to develop innovative genomic products for applications such as functional genomics, synthetic biology, qPCR and PCR, CRISPR, and next generation sequencing, of course. We are also providing our products for other companies. When it comes to NGS portfolio, we offer solutions for many applications with various library preparation kits, different workflows for targeted enrichment or library normalization, everything that would fall between the extraction and sequencing itself. Many of our protocols have scripts available for various automation platforms. This is a very busy slide, so sorry for this. Today, I would like to focus on only one aspect of our NGS portfolio, and namely NGS library preparation methods. Library preparation is a necessary step to turn input DNA or RNA into final libraries before targeted capture or sequencing. I will explain workflow details of our library preparation solutions and discuss various research applications with which our products can help. So let's start with DNA applications and DNA sequencing. The first kit I would like to introduce is called xGen DNA Library Prep Easy Kit, formerly known as 2S Turbo Kit. It has upgraded amplification module and helps to improve library yield, which in turn decreases the duplication rates. This improvement in yield, coupled with the broad input range starting from 1 nanogram of DNA, and also the availability of 1,536 UDI, so unique dual indexing primers. This allows for increased multiplexing capacity for a variety of applications, such as whole genome sequencing, targeted sequencing using hybridization capture, such as whole exome sequencing, genotyping, and variant detection. And it can be also compatible with library normalization. The xGen DNA Library Prep Easy Kit is available in 2 configurations as shown. In both cases, unfragmented genomic DNA is used as an input, and it's then subjected to enzymatic preparation, which includes fragmentation, end repair, and A-tailing. And then the molecules can be subjected for ligation, either with short stubby adapters that would require indexing PCR, or full-length adapters that may not need PCR. So to highlight those 2 different workflows again, the xGen DNA Library Prep Easy Kit, which supports PCR-free library generation, is called UNI, which stands for Universal. And for this kit, the PCR is not needed, as the full-length adapters would already contain indices. While the standard workflow, xGen DNA Library Prep Easy, contains Y-shaped study adapters, and it's required to purchase also UDI primers together with this kit to generate full-length library molecules by indexing PCR. Our recommendation for the unique workflow is to use full-length xGen UDI UMI adapters, and the recommendation for the standard workflow is to use UDI primers. Choice of adapters and/or indexing primers recommended for each library is also represented in this table and highlighted for the EasyKit. I will be showing this table a few times throughout my presentation. Please note that our unique dual indexing primers, UDIs, can be also purchased as compatible with Normalase. Normalase is the enzymatic normalization reagent, which was developed to streamline library concentration balancing. Unfortunately, for the sake of time, we will not discuss the Normalase workflow today. It will be covered during our upcoming webinars. I was mentioning that we recommend full-length UDI UMI adapters for PCR-free workflows, as well as stubby adapters and UDI primers for standard workflows. Those are our off-the-shelf products, but we can also support various custom NGS solutions and adapter options. And the comparison between our stocked products and possibilities for custom products are summarized in this table. So as you can see, full-length UDI UMI adapters, they contain not only unique dual indexing, but also unique molecular identifiers. And they come in certain reaction size and certain concentration, while we allow for a lot of flexibility with custom solutions covering different type of indexing, various shipping containers, and even adjusting for custom index length or sequence. Now I would like to talk about the performance of the kit. The xGen DNA Library Prep EasyKit was benchmarked here in this study against another DNA library prep kit that uses enzymatic fragmentation. Libraries were prepared with both kits and evaluated and were subjected for enrichment with IDT's pan-cancer panel. The xGen DNA Library Prep EasyKit requires a lower number of PCR cycles to generate yields equivalent to the other kit when using the same input material. At low inputs, the xGen DNA Library Prep kit provides more uniform insert size among varied inputs, higher coverage, and fewer PCR duplicates, especially visible for low inputs as 1 nanogram. The kit was also benchmarked against another supplier and tested with 1 nanogram of DNA harvested from a mock bacterial community. The DNA was converted into libraries using IDT, xGen DNA EasyKit, as well as other enzymatic fragmentation library prep kit. The EasyKit provided a greater library yield from fewer PCR cycles and fewer PCR duplicates compared to other suppliers' kits, as supported by the data presented here. From the same data as were shown on the previous slide, here we also looked at the normalized coverage across the distinct GC compositions of the mock community. We see more even coverage across the various GC content windows compared to the competitor kit, which shows a bias toward GC-rich coverage and some bias in AT-rich regions. So to summarize, xGen DNA Library Prep EasyKit is perfect solution for standard sequencing applications. It has improved yield for enrichment, is Normalase compatible. We have pre-plated unique dual indexing primers that enable multiplexing of up to 1,536 samples at the same time. It covers DNA with input between 1 nanogram to 1 microgram. It contains the enzymatic fragmentation module, is the best suitable for samples such as genomic DNA, rather good quality FFPE samples, PCR amplicons, or fresh frozen tissue. The next kit that I wanted to discuss is xGen DNA Library Prep MC Kit, formerly known as 2S Sonic Kit. It is the non-enzymatic fragmentation alternative to xGen DNA EasyKit. Easy stands for enzymatic, MC for mechanical. It requires already fragmented DNA as an input, for example, Covaris-Sure DNA. And similarly to the EasyKit, it supports applications such as whole genome sequencing, targeted sequencing with hybridization capture, sequencing of metagenomic samples, identification of germline inherited CNVs and indels, low-frequency somatic variant identification, or copy number variation in the identification. The streamlined protocol combines end repair and A-tailing into a single reaction and then goes straight into adapter ligation before the first purification is performed. So that allows for a quick turnaround time from DNA to final library. And the flexibility in adapter option enables as well PCR-free sequencing or with the full-length indexed Y adapters, or alternatively, a stubby adapter and the indexing primers can be utilized for applications where PCR is required. All in all, library prep can be completed in around 2 hours, which is comparable to other kits with fast workflows, as presented on this scheme here, comparing to supplier A and B. And the MC kit has the same recommended adapters and indexing primer solutions, depending if the standard or uni version is chosen. It is also compatible with Normalase. And the same as for EasyKit, we offer full-length UDI UMI adapters, as well as stubby adapters and UDI primers as off-the-shelf products. And we can also support custom adapter solutions, similarly to EasyKit. We have also benchmarked this kit against competitors. Here we were generating PCR-free libraries. And as you can see, the library yields were comparable across all 3 kits, as presented in the panel B. And genome coverage at the mean of 10x showed that the xGen DNA MC kit had more balanced relative coverage of high GC regions than kits A and B, as shown here. We have also used this kit to generate libraries with mock metagenome DNA samples. And again, we were comparing using also kits from vendors A and B. And what we see, first of all, is that equivalent insert sizes were generated for all kits. And we see slightly lower duplication rate for IDT kit compared to other kits. In the panel C, you can see representation of the various genomes in the mock population, and it was consistent with the expected percentage, with the expected proportion, showing good representation of genomes with varying average GC content. So as you can see, the expected proportion is here highlighted in dark gray, while the performance of 3 different kits is below, and with IDT is represented in dark blue. So to summarize, the xGen DNA library prep MC kit is a standard DNA library preparation kit that is compatible with already sheared or degraded DNA that doesn't require further shearing. It's streamlined 2-step workflow with optional PCR. It allows for high multiplexing capabilities. It supports input range between 1 nanogram and 1 microgram. And it's the best used for samples such as genomic DNA from tissue, blood, microbial isolates, or environmental samples. I will then jump to next DNA kit, DNA library preparation kit called xGen cfDNA and FFPE DNA kit, formerly called PRISM. And this kit is well suited for low quantity or low quality samples like cell-free DNA or FFPE samples, although it's also compatible with high quality DNA as well. By using a novel method of library construction that also includes built-in unique molecular identifiers, UMIs, the kit allows for both sensitive and accurate detection of low frequency variants from a variety of sample inputs. It can be also used, of course, for whole genome sequencing from degraded samples. A few words about the workflow. So the kit was optimized for low input and degraded samples, and it takes advantage of sequential single-stranded ligation process that utilizes a novel engineered ligase and modified adapters. The enzyme is unable to ligate inserts together, which minimizes the formation of chimeras. due to the proprietary modifications of adapters, also adapter dimer formation is virtually eliminated. Altogether, this strategy delivers a really high conversion rate and high library complexity, higher than conventional TA ligation-based methods. This allows for highly sensitive and low frequency variant detection. In addition, unique molecular identifiers, UMIs, sequences are added to the insert during ligation 1. And this unique single-stranded ligation of fixed UMI sequences enables strand-specific molecular indexing by independently tagging the top and bottom strand. The overall time for library preparation with xGen cfDNA and FFP DNA kit is around or a bit above 3 hours. Currently, the only recommended indexing option for this kit is using our standard unique dual indexing primers, as it requires indexing PCR to generate full-length library molecules. So why are we so excited about this kit? The xGen cfDNA and FFP DNA library prep kit delivers higher conversion rates, complexity as shown. So conversion rates are presented here at the top in panel A. Higher complexity as shown by counting unique molecules at the bottom of panel A. And it also shows higher mean target coverage when compared to kits from 2 other suppliers. We have also used this kit to demonstrate that it's very sensitive for ultra low variant identification. When trying to call a low variant allele frequency variance, for example, below 1%, we observe there is a massive increase in the positive predictive value metric, which is the percent of the variants that are called that are true positives. And we can see this while using UMIs for error correction for the IDT kit. And as you can see, this is massively increased when compared to two other vendors' kits, or even IDT kit, but without using this error correction and just by utilizing standard start-stop deduplication. This is important because when detecting rare variants, sensitivity can be always high, regardless of the method employed, meaning that there will be always true positives will be always visible. The problem is that without error correction, there are going to be a large number of false positives as well that are called with methods that they don't employ a UMI correction for PCR errors, sequencing errors, and DNA damage errors. And those are errors that occur at low levels, but when variants are also present at for example, below 1% levels, they can severely impact PPV or specificity. And here we have also tested the kit on FFP reference materials. So we show that we see increased yield from xGen cfDNA and FFP DNA library prep kit when using FFP reference standards with varying quality, as indicated here. So, and also indicated by the DIN number standing for DNA integrity number. So with the xGen cfDNA FFP DNA kit, we obtain the yield with the poorest quality of FFP sample, similar to that of the best competitor with the highest quality FFP DNA. This yield increase allows for more samples to be processed by passing any applicable QC threshold prior to sequencing. As with the prior cfDNA data that was presented before, we see an increase in library yield, but also that translates into higher unique molecules and target coverage after sequencing. The xGen cfDNA FFP DNA kit provides uniform coverage across the human genome and has a low level of GC bias. And this chart looks at the frequency of GC poor and GC regions across the human genome. Outside the very extremes of GC content, we see normalized coverage is close to 1 across the majority of the genome, meaning that library prep is not introducing any extra GC bias. So in summary, the wide input range and novel method of library construction for the xGen cfDNA and FFPDNA library prep kit provides superior yields from low quantity and low quality samples. This increased yield translates into more samples that can be sequenced and the higher complexity library that can be utilized. And in conjunction with the UMI for error correction, it is well suitable for calling rare variants while minimizing false positives. The input range that is supported by this kit is between 1 to 250 nanograms. When working with good quality DNA samples, they would require fragmentation prior to the library preparation. But of course, the best supported samples for this kit are cfDNA and FFPDNA samples. Now I would like to move to another kit, to next kit called xGen single-stranded ssDNA and low input DNA library prep, formerly known as 1S Plus DNA kit. It supports very low input amounts and has the benefits of utilizing both single-stranded DNA and double-stranded DNA as an input. Key applications for these kits are mentioned here. For example, metagenomic samples, single-stranded DNA samples, heavily damaged DNA. It has been also shown to be used in chromatin studies, in viromics, or in difficult to extract DNA samples or heat-denatured pathogenic samples. The kit is using the adaptase technology. Adaptase technology is a proprietary technology that is employed to add a 3' adapter to single-stranded DNA fragment. So in case double-stranded DNA fragments are also present in the sample, there is a denaturation step to generate single-stranded DNA molecules. And then the adaptase technology adds the truncated 3' adapter to single-stranded fragments. It is independent of the sequence, and because of this addition to single-stranded DNA, it really improves the efficiency of the conversion. Next step, there is extension of the second strand. In the next step, there is ligation of the 5' and adapter, truncated adapter, and the full-length library is generated by indexing PCR. Since truncated adapters are introduced during workflow of xGen single-stranded DNA and low input prep, the kit is only compatible with indexing primers, and it can be also combined with the Normalase. This figure compares here a traditional library prep to xGen single-stranded DNA low input kit and assumes damaged DNA top strand nick as an input. And this is now important to show why it may be advantage of using single-stranded DNA kit when working with damaged or samples that are suspected to be damaged. So as you can see here on the right-hand side in a traditional workflow, if there is a nick present at the top strand, this traditional library will effectively lose information from the top strand that's nicked because only a single adapter makes it into either side of the damaged fragment. After the denaturation during the initial PCR step, the damaged top fragment becomes 2 fragments, but with only single adapter present on the now separated fragment. So it will not amplify or sequence properly. On the other hand, the workflow of the xGen single-stranded DNA and low input kit denatures samples before library preparation, and it can make libraries from single-stranded DNA. This enables the damaged fragment to be properly ligated, allowing for amplification and sequencing of both the damaged top strand and the intact bottom strand. Here we show some internal studies where we have tested xGen ssDNA and low input kit to make libraries from a mixed population of single-stranded and double-stranded genomes using mock metagenomic samples. Not only the library constructs well, the kit constructs libraries well from both types of input, but also the expected and obtained proportions of each genome within the mock mix were retained, as you can see here in these columns, as expected and generated with the xGen ssDNA and low input DNA kit. Another advantage of using this kit is when dealing with difficult to extract metagenomic samples. Often for these kits, there are two methods used, bead beating or sodium hydroxide boiling. The bead beating is less harmful for DNA, however, it is not as efficient and may, even after the bead beating, there is still often analyzed cells present in the sample. Sodium hydroxide is quicker and results in higher yields as compared to bead beating, but it also results in single-stranded DNA. And as such, to make a library from DNA extracted from this method, a kit such as xGen single-stranded DNA is required. xGen single-stranded DNA and low input kit was featured in various studies. And here researchers were using the kit to detect both single-stranded and double-stranded DNA viral genomes obtained from dorm room air filters with no selection against one or the other type. Another example where the kit was used is the famous NASA twin study, where the kit was used to assess changes in microbiota from two brothers. In summary, xGen single-stranded DNA and low input library prep kit is perfect for metagenomic, but as well oncology and liquid biopsy samples, and was also shown to be useful for ChIP-seq studies. It allows for accurate analysis of both single-stranded and double-stranded DNA. Simple workflow is around 2 hours, and because of the adaptase technology, it accommodates inputs as low as 10 picogram. If using good quality sample, it requires fragmentation, mechanical fragmentation prior to use. It was shown to work well with sample types as genomic DNA, FFPE, PCR amplicons, or fresh frozen tissues. Now I will switch over and move to methylated DNA sequencing options. Our xGen Methyl-seq library prep kit can be used for all kinds of applications that use bisulfite converted DNA, such as whole genome bisulfite sequencing, or targeted sequencing with hybridization capture enrichment or RRBS enrichment. It can be also used for genome-wide methylation identification on cfDNA, on single-cell methylation sequencing with the adaptase module, and is also compatible with bisulfite converted DNA enriched by methylated DNA IP, ChIP, and other methods. It is compatible with various sample types, so genomic DNA, FFP, cfDNA, as well as fresh and frozen tissue. xGen Methyl-seq library prep uses introduced earlier adaptase technology, and it uses bisulfate converted DNA as an input. So the bisulfate conversion is happening prior to library preparation. There is adaptase step where the truncated 3' end adapter is added to single-stranded molecules, extension, addition, ligation of the 5' end of truncated adapter, and final step, indexing PCR. So why this is now important, I highlighted that the bisulfate converted DNA is used as an input. This is the difference between the xGen Methyl-seq DNA library prep kit versus the traditional method, where DNA is first subjected to library preparation without PCR amplification, then bisulfate conversion is performed, and then the PCR amplification to generate functional library molecules. The problem with the traditional method is that it often, the bisulfate conversion is harmful for DNA molecules, and it can lead to broken library molecules, decreasing the complexity of the library. Another method often used for methylation studies is random priming, And even though the random priming happens after post-bisulfite conversion, it is also less efficient and more biased compared to xGen metal-seq DNA library workflow. Similarly to xGen single-stranded DNA and low input prep, xGen metal-seq libraries requires amplification with indexing primers, and the kit can be also compatible with Normalase. So to demonstrate the efficient and unbiased performance of the xGen metal-seq DNA library prep kit, we have performed a titration experiment where we have used various input amounts of Arabidopsis thaliana genomic DNA and compared our workflow with two alternative methods, so the traditional method and random primer method. And we were using the same input DNA for each. As you can see, our kit demonstrates higher, more comprehensive genome coverage, lower duplication rates, compared to both alternative methods for each of the input samples. And here I'm just representing the results for 10 nanogram used as an input. The traditional method had equivalent result at the 100 nanogram input, but lost significant complexity with the lower inputs. And in contrast, the random prime method had lower complexity at higher duplicate rates for each input concentration. In the same experiment, we have also looked at the CPX sites uncovered and covered. So the CPX are the CPG and CPH sites where the methylation occurs in plants. Our kit and the traditional method leave a minimal amount of CPX sites uncovered, with some increase for the lowest input amount for the traditional method, while random priming shows elevated percentage of uncovered sites at every input tested. The percentage of CPX sites covered at least 10x is greater than 90% for metal-seq at 100 nanogram, and it decreases to approximately 75% at 1 nanogram. However, it is still superior compared to the traditional method or the random primer method at each input. Without going into detail, I just wanted to mention that xGen metal-seq DNA library prep kit can be also used in reduced representation bisulfite sequencing, or shortly RRBS, and as well can be coupled to targeted enrichment via hybridization capture. Last but not least, I just wanted to show one publication where our kit was compared to two other vendors' kits in high throughput whole genome bisulfite sequencing. And the three workflows were compared based on various sequencing metrics. And as you can see, our kit has scored higher in almost all of the metrics when compared to competitor I and competitor Q. So to summarize, xGen metal-seq library preparation workflow is suitable for input ranges starting from 100 picogram to 100 nanogram. It supports genomic DNA as well as lower quality FFPE and cfDNA samples. It is powered by the xGen adaptase technology. So it reduces workflow while generating less biased libraries. And it is compatible with high multiplexing capabilities up to 1,536 samples. Regarding methylation studies, IDT has one more product to offer. We also provide adaptase technology as a separate module, and the xGen adaptase module can be implemented for single-cell whole genome bisulfite sequencing. It is compatible with single cells that are isolated from fresh or frozen tissue, FACS sorted or laser captured, as well as just with isolated nuclei. This product was developed in a collaboration with a customer, which resulted in a science publication describing the method, for which I'm going to show the workflow here. So in this study, the researchers has dissected brain tissues and isolated nuclei, which were later stained and sorted into single wells. On a single well plate, protein K digestion and bisulfate conversion appeared, and then the samples were subjected to library preparation And I'm just going to expand the last figure. So in this single cell whole genome bisulfite sequencing, bisulfate converted single-stranded DNA were subjected to random priming with primers that would contain truncated 5' end adapter sequences. After purification, adaptase technology was employed to add truncated adaptase stubby adapter at the 3' end of these molecules, and indexing PCR was used to generate full-length library molecules. So now I will finish with DNA library methods, DNA NGS library preparation methods, as well as methylated DNA library methods, and I will switch to RNA as an input. Just before discussing RNA library prep workflows, I wanted to present a reminder that about the RNA inputs used for various RNA library preparation kits depending on the application. Total RNA is usually subjected to poly-A selection for whole transcriptome sequencing or for ribosomal depletion when, for example, information about non-coding transcripts is also required. However, researchers planning to use targeted enrichment with hybridization capture downstream to library preparation can utilize total RNA as an input as well. IDT offers two RNA library preparation kits, xGen broad range and the standard xGen RNA library prep kit. I will start with the xGen broad range kit workflow. First, the fragmentation is occurring, and after that, reverse transcription is performed with random primer. Single-stranded cDNA is synthesized and then subjected to adaptase technology. Because adaptase technology is used here and it uses single-stranded DNA as an input, it does not require the second strand of cDNA synthesis. Upon the adaptase technology, there is extension of the second strand, ligation of the P5 truncated adapter, and the final full-length libraries are completed with indexing PCR. Overall, it is around 4.5 hours of hands-on during this library preparation. xGen RNA library prep kit is very similar with following differences. The random primer used for reverse transcription already contains truncated adapter, P5 adapter sequence. In that way, the cDNA that is synthesized during this step already contains this adapter at the 5' end. Then adaptase technology is used to add the 3' end truncated adapter, and it is followed by indexing PCR to generate fully functional index libraries. For both kits, and similarly to other kits that employ adaptase technology, indexing primers are used for generation of the functional library molecules, and the kits can be followed by normalization with Normalase. Oh, I'm sorry. Because of the adaptase technology, xGen library prep kit generates minimal adapter dimer formation, even for low input samples, and does not require additional adapter titration, as it is the case for other vendors' kits, in this case compared to vendor N. And as you can see, even 100 picogram of mRNA was used for this purpose, and no dimer, adapter dimer formation was observed. When it comes to handling and incubation times, our xGen RNA library prep kit is also currently one of the fastest on the market when it comes to the preparation. And here we are comparing it with two very commonly used kits, RNA library preparation kits from suppliers K and I. Here, this is a very busy slide highlighting differences between both of the kits, the xGen broad range RNA library prep kit and the standard xGen RNA library prep kit. I just wanted to highlight two things. First of all, the input quantity. The broad range, as the name suggests, can really be applied for various diverse samples with a wide range of input amounts. And while xGen RNA library prep kit is less sensitive, and it can support only as low as 50 nanogram of total RNA or 1 nanogram of mRNA. It is the turnaround time for this kit is currently the fastest on the market. Both of the kit, they use the adaptase technology for tailing and the ligation of the first strand cDNA on the first strand cDNA, and both of them, they are capable of multiplexing up to 1,536 samples with our unique dual indexing primers. So just to summarize, the broad range RNA library prep kit is more suitable for broad input amounts. It generates fewer adapter dimers compared to other vendors' kits. It's ideal for diverse samples, while the xGen RNA library prep kit, it has fast workflow. It is a cost-effective option when good amount and quality of RNA is available. When it comes to performance of the two kits, we have performed some benchmarking studies. Firstly, for the xGen broad range RNA library prep kit, we compared the coverage across transcripts length for 2 different input amounts and between 3 different kits, IDT kit, supplier N and K. As you can see, we obtained fairly similar evenness of the coverage for all the kits, but please note that we have used 10 nanogram of universal human reference total RNA for all kits, but for the lower amount, we have used 100 picogram of standard mRNA sample only for IDT and the kit from supplier N, while the supplier K required at least 1 nanogram of mRNA. Next, we have looked at expression correlation between samples with decreasing input amounts. Here we have used 100 nanogram replicates and generated libraries and look how the replicates correlate. And then we have compared these results with expression studies looking at this the expression analysis obtained with 50 nanogram and 10 nanogram of the human reference total RNA sample. As you can see, there is really high expression correlation even at the very low input amounts. We have done the same, but with the standard xGen RNA library prep kit. Here we were using 500 nanogram for 2 replicates, and then the decreasing amounts of inputs were used as depicted here, 100 nanogram and 50 nanogram. And again, we were correlating these results with high input used for library preparation. And again, we obtained pretty high expression correlation. The xGen RNA library prep kit was also tested against other vendor kit from supplier K and tested for the mapping rate and for the strandedness. So as you can see, we obtained higher mapping rates when compared to supplier K and measured by the percentage of mapping rate for various input amounts. The kit, xGen RNA library prep kit, also maintains higher percent of strandedness when compared to this particular competitor. In different study, we have used our kit as well as supplier K and N and measured mapping rates, number of genes detected, number of transcript detected, and percentage of duplicates. For all tested input amounts, the xGen RNA library prep kit performed better than kits from vendors N and K, giving higher mapping rate, higher number of detected genes and transcripts, and a lower or comparable duplicate rate. Lastly, we have tested the performance of our kits on low quality FFPE RNA samples. The RNA libraries were prepared with the xGen broad range kit and subjected to hybridization capture with the exome panel. We have analyzed the library yields, the mapping rates, and various other metrics, and we can see that for all samples, so standard sample as well as FFPE RNA samples, we obtained high yields, high mapping rates, high exonic rates across both intact and damaged samples. So to summarize, both of our kits for xGen RNA library preparation, broad range and the standard RNA kits, they are suitable for applications such as gene expression profiling, identification of alternative splicing, identification of rare and novel transcripts, gene fusions, isoforms, structural variants, for analysis of allele-specific expression, detection of disease-associated CNVs, and they are both compatible with hybridization capture enrichment downstream of the library preparation. When it comes to sample types, broad range, as mentioned before, is suitable for really wide range of quantity and quality of RNA, starting from high quality RNA from blood, saliva, high quality FFP RNA, as well as degraded FFP or low input amounts, while standard RNA library prep kit with faster workflow, it is more suitable for rather good quality RNA and also good quality FFP RNA. So with this, I would like to thank you for your attention, and I will be happy to take any questions.
Thank you very much, Julia. Okay, so we have one question. The question is, can you comment on the approach, workflow, or kit which you would recommend if you're interested in sequencing both the DNA and RNA of a sample on the same sequencing run?
We at IDT, we do not have one kit that would be suitable for converting both DNA and RNA within one tube to generate functional library molecules. What we could recommend is maybe using two different kits, or depending on what's the sample type from which this DNA and RNA, these nucleic acids were isolated, to use, for example, prior to, so one can use any of our standard DNA library preparation kits and RNA library preparation kits. It would be also possible for many of the DNA kits that we are using to convert RNA first to cDNA and then use it as an input for DNA library preparation kits. But now this really depends what is the sample type, what's the aim of the sequencing, is it looking at specific regions? Because I mean, sequencing both RNA and DNA in one sample, one has also needs to consider, for example, the coverage and depth that it's aimed to be achieved with sequencing. So it is not always that easy to just combine samples together and sequence them, as, for example, RNA libraries versus DNA libraries may require different sequencing considerations.
Okay, so if you have more information on that, for the person that posted that question, if you require any more information, please feel free to reach out to us at Whiteside, and we can try and assist you more once we have some more details from you. We have another question. Does IDT have any NGS prep solutions for nanopore sequencing?
No, currently we support only libraries prepared for short-read sequencing at Illumina platforms. However, depending on the application, we can provide some of the modules of the workflows for long-read sequencing. So we do not have any NGS library preparation kits, but we are very happy, and we often provide solutions for hybridization capture that utilizes libraries for example, ONT platforms.
Thanks, Julia. And what are the pros and cons of the EZ over the MC, or the MC over the EZ workflows?
The EZ and MC kit, maybe I will go back to this slide. So the EASY and MC kit are actually very similar to each other in terms of workflow. The EASY kit, the difference between them is that the EASY kit contains the enzymatic fragmentation, while the MC kit does not contain enzymatic fragmentation module. Means that if there are researchers who would like to use or they already have sonicated or naturally fragmented DNA with good quality, and they would like to use it for DNA library preparation, we would recommend the MC kit. If there are researchers who do not have access to sonicate or who have good quality unfragmented genomic DNA, then we would recommend the EASY kit. In terms of specifications, what's the input amount that can be used, the workflows, using PCR-free or PCR amplified libraries, they are the same in that sense.
Okay. All right, we don't have any other questions right now. Let's just see if anything pops up. Okay. Julia, thank you very much today. We really appreciate your time, and thank you to all the attendees for spending some time with us this morning, learning a little bit more about the library prep kits from IDT. And yeah, just you can keep an eye out on social media or anything. If you have not registered for the other 2 webinars, it is also on the same newsletter as the invitation to the first webinar. But if you don't have that, feel free to reach out to us. We'll be happy to send that on to you if you would still like to register. Okay, so we don't have any other questions, so I think that's it. Thanks, everyone.
Thank you very much.
Bye.